Good morning, students. This lecture is from paper 205 and the chapter is Pure Culture Techniques. In that, today I'm going to talk about nutrition, like how do you decide the nutrient composition of a media. The flow of lecture is something like this: introduction and various components. How do you decide the various components and what are the various components, and what is the composition of the various components, and factors which affect growth, like other physical requirements with the composition which you need to provide, and then different types of media what is required. This comes in fourth chapter of paper Microbiology. So we'll start the lecture. So we'll define what is culture media. Culture media is something you give a special mixture of all the requirement of a cell, using which the microorganism, either the bacteria or algae, grows happily in that nutrients what you have given. So a culture media is a special media used in microbiological laboratories to grow different kinds of microorganisms. So it should contains all the requirement of the microorganism. So there are different types of microorganisms, right? Which has unique properties. That means they have a different requirements. Some they grow happily everywhere, they're ubiquitous. Some they are very fastidious. They grow in particular required places. Like some they grow in extreme temperature. Some they grow in a halophilic environment. So every organism is different. So we'll broadly understand the different requirements of microorganisms. So basically you can divide the whole group of organisms based on the way they synthesize their food in terms of autotrophs and heterotrophs. So autotrophs, you know that they derive energy from non-organic carbon sources, and they synthesize their cellular molecules from carbon dioxide and nitrogenous compounds. Is that much enough? And then using in that also you have two groups. The one which generate their food using the sunlight are called as phototrophs, and the one which require some inorganic compounds to build their cellular molecules, but there is no absolute requirement of sunlight, are called chemotrophs. And you have heterotrophs which require organic compound. Like you can see in this flow diagram, basically based on energy you can divide into two groups. The one which completely depend on the light are called phototrophs. The one which depends on the chemical form of energy are called chemotrophs. So in phototrophs also, where the organisms require organic compounds, they are photoheterotrophs. The one which can survive only with carbon dioxide are photoautotrophs. In photoautotrophs, again use water to reduce oxygen. Such a group of organisms are called oxygenic photosynthetic microorganisms. Cyanobacteria is one huge groups under this. The one which does not require water to reduce carbon dioxide are called anoxygenic photosynthetic bacteria, basically a huge group, a green or purple bacteria. In in the group photoheterotrophs, you have the sulfur bacteria, green non-sulfur bacteria, purple non-sulfur bacteria coming under this photoheterotrophs. And the one chemotrophs, again you have two groups, chemoheterotrophs and chemoautotrophs. Chemoautotrophs are, they don't require any organic compound. They can use just the atmospheric carbon dioxide and then synthesize their major requirements. Examples under chemoautotrophs are hydrogen, sulfur or iron, nitrogen or carbon monoxide oxidizing bacteria. In chemoheterotrophs, if the final electron acceptor is oxygen, where most of us, all animals, fungi, protozoa belong. They come in one category. Another groups in chemoautotrophs which do not require oxygen, they grow in anaerobic region, but the terminal acceptor of electron will be an organic compound. Then you have the streptococcus group. If it is inorganic compound, you have the clostridium group. A few examples. So here, by seeing the chart how they are divided, only you can imagine what are the requirements of the bacteria. Like similarly you can divide other groups also. So basically, if you see the elemental composition of a cell, then you have the higher elements. The majority of the elemental composition is based on oxygen, hydrogen, carbon, nitrogen, phosphorus, and sulfur. Other elements which are less in amount are potassium, calcium, iron, sodium, and there are trace elements also which are very much required for the enzyme function of microorganisms. So based on this elemental composition, you can divide the nutrient media composition components as major mineral nutrients, minor or micro mineral nutrients, trace elements. The another group is the growth factors. And with all this, what are the physical requirements which needed for an optimal growth of microorganism? The first group is major or macro mineral nutrients. So now when you take any cell, until unless they are viruses, all the cell contains cytoplasm where most of the activity happens of a cell. That means the cytoplasm is made up of water. That means the total cell volume, 80 to 90% total weight is of water. So the major requirement should be the water, that is the hydrogen and oxygen. The solid matter of cells contains, in addition to hydrogen and oxygen, the other macro elements like carbon, nitrogen, phosphorus, sulfur, potassium, magnesium, sodium, calcium, iron in decreasing order or abundance. So total dry weight of the cell, we can say oxygen, hydrogen, carbon, nitrogen, phosphorus, and sulfur are required. So where, and this is a chart which tells where the carbon is required, oxygen is required. Like see, carbon, it is in all organic compounds, carbon is a major constituent. So if you see the right from the sugar to the nucleotide or the lipid in the membrane, everywhere you can find carbon. So the percentage of dry weight of carbon is 50%. The rest 50% is divided under other major mineral nutrients. Followed by carbon is oxygen, where oxygen is in the form of water or organic cell material or molecular oxygen, which serves as electron receptor in aerobic respiration. Followed by that is nitrogen, like most of the nucleic acid, proteins, coenzymes contain nitrogen. Followed by that is hydrogen, where the cellular water organic cell material contains hydrogen. Followed by that is phosphorus, which is required, which you can see in nucleic acid, phospholipids, and coenzymes. Followed by that is sulfur, the major amino acids which require sulfur, and some coenzyme also require sulfur. Followed by that is potassium, sodium, calcium, magnesium, and iron. So iron is required in cytochromes or other heme or non-heme protein as a cofactor for number of other enzymatic reaction. So the next group of requirements is minor or micro elements. So here in general do not use only microorganisms do not use only macro elements, but they also require other elements like cobalt, copper, manganese, molybdenum, nickel, selenium, tungsten, vanadium, and zinc, which are required in not a high percentage but in residual fractions. So these are referred to as micronutrients or sometimes trace elements. So they are very critical for the function of the cell because they play important role in cells catalysis, and many of them have structural roles. For example, diatoms require silica which they put in their cell wall. There is a chart, you can see the micronutrient and their cellular function. There are various enzymes which require these elements as cofactor. The next group is the growth factors. The growth factors, basically you have three types. Here most of the microorganisms, they can synthesize their own other required factors, but in general there are three groups which are required. Some of the amino acids, for some microorganism you need to supplement with purines and pyrimidine. For some microorganisms, some type few groups of vitamins should be supplemented in the media for their best optimal growth. So these are the vitamins and their functions. Like riboflavin is required as it's a precursor of flavin mononucleotide and flavin adenine dinucleotide. Cobalamin is important for reduction of and transfer of single carbon fragments and synthesis of deoxyribose. Para-aminobenzoic acid is a precursor of folic acid. Gut bacteria can synthesize folic acid, but when you're growing that, you need to supplement with the precursor, or certain bacteria they require folic acid in their medium. Vitamin B6, which is required for amino acid and keto acid transformation. So this was the brief glimpse of what are the major micro and trace element requirements. Next we'll go to the physical requirement or the conditions in which you have to maintain for the optimal growth. We'll take bacteria as the group and then start describing, and it holds good. May not all the groups, like for temperature, mostly fungi, if you take, they are mesophilic. So may not all the categories, but most of the microorganisms fit into these groups. So temperature is the very important criteria for the growth of microorganism. Once you have defined the media components, and then these are the conditions. So bacteria has minimum, optimum and maximum temperature for growth, and it can be divided into following groups. Psychrophiles are cold loving bacteria. Basically it can grow between -5 degrees to 15 degrees. They are usually found in Arctic and Antarctic region. Mesophiles are like optimum temperature, somewhere between 25 and 45 degrees is the optimum growth. Most bacteria are mesophilic, and this include common soil bacteria and bacteria that live in on the body. Thermophiles are some heat loving bacteria. They have an optimum growth temperature between 45 and 70 degrees and are commonly found in hot springs and in compost. And hypothermophiles are bacteria that grow in very high temperatures, and their optimum temperature range between 70 and 110 degrees. They are usually members of archaea and are found growing in the hydrothermal vents and in great depth. So here is a small chart which tells the psychrophiles which grow at lower temperature, showing optima at 4 degrees. For example, Polaromonas vacuolata. The mesophile example is Escherichia coli. Thermophile, Bacillus stereothermophilus. Hypothermophile, where it Temperature optima is little higher than the thermophile. Example is Thermococcus celer. Hypothermophile, another example which needs more than 100 degree is Pyrolobus fumarae. So there is one more chart which shows minimum, optimum and maximum temperature which are required, and these are the few example. Not only general non-photosynthetic prokaryotes, you have photosynthetic bacteria also which can grow at little higher or have higher temperature as optimal temperature for growth, like Cyanococcus exmeos. So followed by this is oxygen requirement, the major requirement. And here also you can subdivide the organism into various groups like obligate aerobes. These are the group which grow only in the presence of oxygen, and they obtain their energy through aerobic respiration. Aerophile is something like if the oxygen is present in low concentration, they can happily grow. They are not inhibited when oxygen concentration is low. But when the oxygen concentration increases about 10%, then the growth is inhibited, because they obtain their energy through slight aerobic respiration. Obligate anaerobes are the organisms that grow only in the absence of oxygen, and they are killed by the presence of oxygen. Aerotolerant anaerobes are like obligate anaerobes which cannot use oxygen to transform, get energy, but they can grow in the presence of oxygen. Facultative anaerobes are organisms that grow with or without oxygen, but generally grow better with oxygen. They obtain their energy through aerobic respiration if oxygen is present, but use fermentation or anaerobic respiration if it is absent. So the next factor is pH. Microorganisms, they can grow in all the three pH scales, that is acidic range, basic range and neutral. Most of them are most of bacteria, they fall in the neutral range and slightly acidic and slightly alkaline, not the extremes. Neutrophiles are something between the range 5 to 8. Acidophiles are some which are growing best below 5.5. Alkali are something which can grow best at the pH above 8.5. Here is a scale which shows what are the examples of the raw materials which you can use for growing acidophiles or alkalophile. Like we can use tomatoes which show the pH below 5 for growing an acidophile, or we can use alkaline natural soil for growing an alkalophile. Pure water has neutral pH. So apart from pH, the ionic concentration is very important, as that determines the water flow in with the organism or outside the organism, moving out of the organism. So that is through the process of osmosis. Osmosis nothing but the movement of the water from its regions of higher water potential to a lower water potential. So basically concentration of water molecules. So this flow is basically the potential energy which drives the flow, and there's a lot of energy which is invested when this kind of changes happen. So the conditions of the media should be properly maintained. uh, so the kind of uh, environment what we can give is like three types: isotonic or hypotonic and hypertonic. Most bacteria require an isotonic environment, or some grow in hypotonic environment for optimal growth. Organisms that grow at relatively high salt concentration are said to be osmotolerant, and they require high salt for their growth also, like some archaea, that require high sodium chloride concentration up to 20%. So higher water potential, less of solute is like the pure water, where water activity is highest. The example of organisms which can grow in such kind of conditions is bacteria and spirulum. Human blood has zero point 0.9 water activity. Streptococcus, Escherichia coli, and most of the mesophilic bacteria can grow in such condition. When you have like halophilic bacteria, or where the ionic concentration is very high, as you see in candy or dried fruit, where the dehydration is happen, the water potential there is 0.7. In such kind of condition, either the fungi will grow very nicely, or organisms like Xeromyces bisporus can grow in such condition. So when the salt concentration is less and high water potential, the best growth is of a non-halophile. As you increase the salt concentration, you get the halotolerant halophile and extreme halophile. Halotolerant example is Staphylococcus aureus, halophile Vibrio fischeri, and extreme halophile is Halobacterium salinarum. So till now what we have covered is knowing the requirement of the different groups of bacteria. Based on the requirements, the groups, you can design the growth conditions of a bacteria. And while designing the media components, what are the major components, what are the micro requirements we came across. And then other than that, we came across what are the physical requirements, like what temperature it should be kept, should be the oxygen requirement, and what should be the pH, osmolarity. All this are the basic important things which you have to keep in mind when you're designing certain media or growth condition. So for growing optimum growth of a particular type of bacteria, so this kind of the media, when you prepare the culture medium, comes in various forms or various types based on the requirement. So that depends on the microorganisms. So when we are adding components, we know that the major requirements are carbon, hydrogen, oxygen, nitrogen. So you have certain chemicals which can be supplied. Like for the carbon requirement, you have glucose, or a complex mixture also can be supplied in the media, like peptone, yeast extract. They also can act as carbon source. For hydrogen requirement, water and other organic compounds are enough. For oxygen, water, oxygen itself, gas, and other organic compounds are enough. For nitrogen requirement, we add ammonium chloride or potassium nitrate. Organic form of nitrogen is like amino acid, nucleotides, and other nitrogen containing compounds can be added. For potassium requirement, KCl will add. Magnesium requirement, magnesium chloride and magnesium sulfate. Sodium requirement, NaCl. Iron requirement, ferric chloride, ferric sulfate, etc. Can be added. So using this, and you can prepare different compositions of media. So you can divide the types of media based on the consistency what you prepare, or based on the composition, or based on the purpose or functional use. So first we'll go up across based on the consistency. That means whether it is a liquid culture medium, or semi-solid or solid medium. Liquid broth, for example, the normal Loria broth, or methyl red Vogus proscaver broth. These are like, you have the components like the major and the minor requirements of the organisms, but you do not have the solidifying agents like agar or gelatin. Semi-solid medium is where you have added the agar but at a very low concentration. They form like soft custard like a consistency. And this kind of semi-solid media are utilized in the cultivation of microaerophilic bacteria or determination of bacterial motility. The extensively used medium is a solid medium which contains the solidifying agent like agar, where the percentage can go maximum up to 2% and are mostly inert solidifying agents. So solid medium has typically physicals. You can use that for understanding the colony characteristic, and then isolating a bacteria for understanding the other features of the bacteria. You can use that media. Next type of classification of culture media is based on the composition. When you know all the chemical components, like you have added NaCl, you have added ammonium chloride, for carbon you are added glucose, for adjusting the pH NaOH or KCl or KOH, when you know the composition of the media very well defined, such is called as a chemically defined media. The chemically defined media can be as simple as I told or as complex. When you have biofuels media, you can supply a complex supplement into the media where the nutrient requirement or the selecting factor can be included. Non-synthetic media or chemically undefined media will have some component like peptone or tryptone, which are added into the media, where the exact composition of that peptone or yeast extract may not be known. So for example, what we prepare nutrient broth falls into that category. So classification based on the purpose or functional use or application. Like, many special purpose media are needed to facilitate recognition or enumeration and isolation of certain types of bacteria. To meet these needs, numerous media are available, like general or basic media. Basal media are basically simple media that support most fastidious bacteria. Examples are peptone water, nutrient broth, and nutrient agar, and they are considered as basal media. This is basically used for isolation. Followed by that, if you want to enrich one particular kind of growth forming bacteria, then you need to use enrichment media, where you will add certain growth factor. Like you have blood agar. Staph you can recognized under the group mixed culture, where you can prepare by adding sheep blood in the percentage 5 to 10% of blood can be added to it, to the culture medium. So then you can know the streptococci, different types of hemolysis. You can further divide the streptococci also. Chocolate agar is also known to be used as enrichment medium, where the chocolate color may be because of lysed blood in the medium. Followed by that, you have other group, that is the next is selective or enrichment medium. Selective medium is usually the solid one, or enrichment medium are the liquid in consistency. They are used or designed to inhibit unwanted commensal or contaminating bacteria and help to recover pathogen from a mixture of bacteria. Any agar media can be made selective media by addition of certain inhibitory agents that don't affect the pathogen of interest. So you can have various approaches to develop a selective media by adding antibiotics or some chemicals, some alteration in the pH or combinations of these can be used to prepare selective media. Examples of selective media I have given here: Thymer Martin agar, which is used to recover Neisseria gonorrhea. It contains antibiotics like vancomycin, colistin, and nystatin. And Lowenstein Jensen media to recover MTB, Mycobacterium tuberculosis, used as a selective media by incorporating a malachite green. Crystal violet blood agar, used to recover uh, pyogenes, which contains crystal violet. And TCBS agar, which is thiosulfate citrate bile salt sucrose, where Vibrio cholerae uses it's used to isolate Vibrio cholerae from fecal specimens which has elevated pH, and which inhibits most other bacteria. Followed by this is the enrichment medium. As I already told, it's liquid culture. It is the broth medium, and here the liquid that also serves to inhibit commensal in clinical samples. Examples of enrichment culture medium is selenite F broth, tetrathionate broth, and alkaline peptone water. Next group is differential or indicator medium, uh, where certain media are designed in such a way that different bacteria can be recognized by just looking at their colony color. Various approaches are used by incorporating dyes or metabolic substrates which are synthesized by those bacteria, uh, because they, once they utilize, they appeared colored colonies. So the media is called differential or indicator medium. Differential media allow the growth of more than one microorganism of interest, but with morphologically distinguishes colonies. Example is mannitol salt agar, where mannitol fermentative organisms give yellow color. Blood agar gives hemolysis of streptococci, where you can differentiate between alpha, beta, and gamma hemolysis. McConkey agar, which gives pink colonies for non lactose fermenters, and pale color for uh, pink color for lactose fermenter, and pale color for non lactose fermenter. TCBS agar, as I already mentioned, thiosulfate citrate bile salt sucrose containing agar, where Vibrio cholerae produces yellow color colony due to fermentation of sucrose. The next group is transport media. Suppose you are isolating some organisms from a clinical sample, it has to be transported to the lab. During that transport, drying can happen, and then you can have commensal organisms which outgrow the required organisms. So certain medias are there, for example, Pike's medium, which is used to transport streptococci from throat samples, or a Carre-Blair transport medium, which is used to transport feces from suspected cholera patients. So this is an example of transport medium. Also, you can know the oxygen requirement of certain group of organisms by using anaerobic media. Anaerobic media is prepared by the required component basal, macro, and micronutrients, and then removing oxygen. If you are using a broth culture, boil nicely so that the oxygen dissolved is expelled out, or add something into the medium so that the oxygen requirement becomes very less, or it creates a reduced environment, like adding iron filling into the medium creates a reduced environment. So example of such medium is Robertson cooked meat medium, where you have this medium is optimally used for growing Clostridium species. Thioglycolate broth, which contains thioglycolate glucose cysteine, is also a good medium, and it is used for knowing whether the organism requires the oxygen or not. Methylene blue is like color indicator, which is like under reduced condition, methylene blue is colorless. When it becomes more oxidative, then there is color change. Apart from this, there are various assay medium where you can screen the requirements of vitamins or amino acids selectively, and then identify what is the composition of a medium for a particular type of organism. Also, you can have antibiotics as a assay medium. Other types of media classification is media which is used for enumeration of bacteria, uh, or maintenance media which is used to maintain the uh, cultures. So these are the references. I took most of the content from Prescott's microbiology. Thank you.